Hello to everyone. In this presentation I would like to share with you a very simple but powerful protocol which is the PCR colony screening. Until some years ago, in the standard cloning approach, after the transformation, some colonies were selected for each plate, inoculated in LB media and grow for plasmid extraction, and then the purified plasmid were subjected to PCR screening to verify the correct insertion of the fragment. But the development of a new formulation for the tech polymerases make possible to perform the PCR screening directly from the colonies. And this reduces a lot the number of plasmid extraction because we can purify and send to sequencing only the plasmids that were positive at the PCR screening. First of all, to perform the PCR screening, we have to choose the primer for the PCR. The primer can be designed to anneal on the flanking region or in the insert, and depending on the primer design, we can just check the size of the insert, the specificity of the insert, or also the direction of the insert. The protocol is really simple, so once you have the agar plate with your colonies, you can prepare a master mix containing the TAC that you will directly use for the PCR and the primers. There are available on the market several nice TAC polymerases that work very well for the colony screening. Those TAC generally contain already the loading dyes who are colored, and you can load directly your sample on the agarose gel after the PCR without adding the loading dye. Once that the master mix is ready, you can distribute it in a multi-well plate for the PCR, pick the colonies one by one and replicate in a twin plate and inoculate it on the PCR reaction. Then you can cover the multiplate with a specific foil and around at the end of the PCR the sample can be loaded directly on agarose gel to check which colony are positive and which colony are negative. And the plate with the replicates could be used to inoculate the positive clones for plasmid extraction. Looking to the price of the TAC, you can think that this protocol is expensive, but you have to take in mind that this protocol allows to the scientists to save time and to reduce the number of plasmid extraction, and plasmid extraction require money and time. So in conclusion, I think that PCR cloning screening is very intriguing protocol, especially for manual cloning. I would like to say a great thank you to Cecilia Brettoni that in 2009, during my time in Novartis, helped me to understand that if you are well organized with this protocol, you can really screen a huge number of clones in parallel with limited effort. Thank you to everyone for the attention. Bye bye. Ciao ciao a tutti. Ciao.